Collyer.
Hi, my name is Janice Lee.
I'm Megan Goins.
And I'm Lyndsay Oakeson, and this is our short and sweet project of DNA replication. DNA replication occurs in a semi-conservative manner, meaning that the template strand is separated and a new strand is synthesized for each single strand of parent DNA. Newly formed DNA is going to have one parent strand and one daughter strand.
DNA replication in prokaryotes starts at the origin of replication or ORC region. The ORC region has 5 sites where DNA A protein will bind, and also a DNA unwinding element, which is an AT-rich sequence that will melt. These 2 elements are cis-acting, present on the DNA at the ORC, and is where the chromosome will assemble.
DNA A protein binds to the DNA A binding sites. This large DNA A complex acts to unwind the DNA unwinding element. Next, DNA B, which is a helicase, binds to the single-stranded DNA in the melted region with some help by DNA C. This complex is the pre-primosome.
DNA C leaves the complex, and the hexameric helicase, DNA B, stimulates binding of the primase, DNA G. Now the primosome assembly is complete. The primosome will stay with the replication with the replosome during elongation at the replication fork. DNA B will act as the helicase to unwind the replication fork.
Single-stranded binding protein binds to prevent the 2 strands from reannealing. The clamp loader recognizes the replication fork and recruits a beta clamp dimer to the primer template DNA. The beta subunits clamp around the DNA, which alters the site on beta and increases affinity for the core polymerase. The tau subunits of the clamp loader bind to the core polymerase to the clamp. There are 3 core polymerases with 3 sliding clamps, and 1 clamp loader, which makes up the DNA polymerase III holoenzyme. Having 3 core polymerase increases the binding efficiency. The clamp loader includes 3 tau subunits, which hydrolyze ATP and bind to the core polymerase, beta clamp, and template strand. The delta subunit opens the beta clamp, and delta prime regulates the delta. The chi subunit binds a single-strand binding protein, and the psi helps connect chi to the clamp loader. The DNA is laid out to illustrate the trombone model, where the lagging strand is looped around so that DNA can be synthesized in the 5' to 3' direction. This trombone model keeps the tau subunits closer together and is a more efficient process.
DNA primase adds an RNA primer to create a 3' hydroxyl in order to start synthesis. The beta clamp and core polymerase start adding nucleotides in the 5' to 3' direction on the leading strand, which can be synthesized continuously. On the lagging strand, the DNA must be synthesized in the short Okazaki fragments because DNA must be made in 5' to 3' direction. This method of DNA replication is called semi-discontinuous.
As DNA synthesis continues, the helicase is going to unwind the replication fork. The leading strand will continue being continuously synthesized straight through, and on the lagging strand, more Okazaki fragments will be made. Wait, who wants a primer?
Oh, I really like primers.
You don't want a
thank you.
nucleotide?
Ooh, I'll take a beta clamp for sure.
Come on, let's show some love for nucleotides, guys.